All right, so you guys remember a few weeks ago, I was kind of freaking out about that tick I found? Well, let me tell you, it sent me down a rabbit hole researching Lyme disease.
Oh, yeah.
Yeah, and it turns out it's way more common than I thought. And the science behind it is, wow, seriously mind-blowing. So today, on our deep dive, we're gonna go full Sherlock Holmes.
All right.
We're gonna investigate how Borrelia burgdorferi, the bacteria behind Lyme disease, actually does its thing. We're talking surviving inside ticks, then getting into our bodies, the whole shebang.
It's pretty wild how this spirochete, this is a fancy word for a spiral-shaped bacteria,
Right.
has developed these like really sneaky survival strategies. It's like this tiny little organism read the book on how to outsmart the human immune system.
Totally. And one of the first things I learned about are these things called plasmids. They're basically these like little packages of DNA that bacteria carry around.
Right.
But here's where it gets weird. Borrelia's plasmids are different. They're both linear and circular, which is super unusual. It's like finding a square basketball.
Yeah.
It throws you for a loop.
They are not your average plasmids, and they're not just oddly shaped. They're like critical for the spirochete to survive. Think of them like USB drives, carrying these special tools that the bacteria needs to, survive and spread. And get this, a lot of the genes on these plasmids are unique to Borrelia,
Oh, wow.
which is a big part of why treating Lyme disease can be so tricky.
So it's like Borrelia has its own like secret stash of weapons that other bacteria don't have access to. That's cool, but also kind of terrifying.
Exactly. And for a long time, we could only guess what those weapons did. But then scientists figured out how to use genetic manipulation techniques.
Really?
Yeah, they could like hack into the bacteria's code.
Wait, hold up. They can actually manipulate individual genes in this bacteria. That's crazy advanced.
It is, and it's incredibly valuable. Now, we can pinpoint which genes are responsible for, the spirochete's ability to do things like infect ticks, infect us, evade our immune systems, even spread to different tissues.
Wow. That blows my mind. So we've gone from barely understanding these plasmids to basically having a map of Borrelia's toolkit. But if we can map it, can't we just shut it down?
Well, that's where things get even trickier, because Borrelia has another trick up its sleeve. It can change which tools it uses depending on where it is.
Oh, no.
We call this global gene regulation, and it all hinges on these fascinating things called alternative sigma factors.
Okay, sigma factors, that sounds intense. What are we talking about here?
Imagine a team of stage managers.
Okay.
They're responsible for changing the set and costumes for a play,
Right,
depending on the scene.
right.
That's kind of what sigma factors do for Borrelia's genes. They help the bacteria adapt to different environments. So by switching genes on or off as need.
Okay, that analogy actually makes sense. So when the Lyme disease spirochete goes from a tick's gut to my bloodstream, it's like a whole new scene.
Exactly.
And these sigma factors are making sure it has the right outfit on.
Exactly. And the two main stage managers for Borrelia are ARPON and RPOS.
Okay.
And they work together, kind of like a relay race, to control which genes are active at any given time.
Okay, I'm following so far. But what exactly are ARPON and RPOS doing? Like, how do they actually switch genes on and off?
Good question. So let's say our little Lyme disease spirochete is chilling in a tick just minding its own business.
All right.
It's a pretty comfy life for bacteria.
Yeah.
Warm, sheltered, plenty of food.
Yeah, it sounds like a pretty sweet deal for a little microbe.
Right. But then the tick latches onto, let's say, you,
Oh.
looking for a blood meal.
Okay.
And suddenly the environment for our spirochete changes drastically.
Oh, no, that's my cue. So what happens to the bacteria when it senses that change?
This is where ARPON steps in.
Oh.
It's like the lookout, sensing danger. As soon as the environment changes, maybe the pH shifts, the temperature drops,
Right.
ARPON springs into action, sends a signal that says, hey, RPOS, time for a costume change.
So ARPON is the first responder. So I'm guessing RPOS is the one who actually makes things happen.
You got it. RPS swoops in and starts flipping switches, turning on specific genes that help Borrelia survive in this new, harsher environment.
Wow. That is seriously impressive teamwork. And that's how Borrelia manages to transition from a tick to a human.
That's a big part of it. But hold on, it gets even more fascinating.
Oh, no.
You see, there's another player in this game of genetic control, a sort of mastermind behind the scenes.
Wait, there's more. I knew this little bugger was sneaky. Okay, you've got to tell me more, but I think we need to save that cliffhanger for part 2. Okay, so last we left off, our Lyme disease spirochete just hitched a ride on me from a tick, and its little genetic control system is going into overdrive thanks to ARPAN and RPOS. But you're telling me there's another player, like a mastermind even. Spill the tea.
Well, this is where it gets next level. We're talking about BOS-R, another transcription factor. If ARPAN and RPS are the stage crew, BOS-R is like the director calling the shots, deciding which scene gets the most attention, how the lighting should be, the whole deal.
So it's even higher up the chain of command than RPUS. That's wild. What exactly is BOS-R doing?
It turns out BOS-R has this, like, particular fascination with RPUS. It's like its star performer. But instead of just saying go or stop, BOS-R fine-tunes RPUS's activity. It's incredibly precise control over which genes get turned up or down.
Wait, hold on, back up a sec. Why does this even matter? Is this level of control, like, common for bacteria?
It's not. And that's what makes Lyme disease so challenging, this fine-tuning. It's how Borrelia stays one step ahead. Think about it. Our bodies are constantly trying to fight off invaders, right?
Right.
Our immune systems have all sorts of tricks up their sleeves. But because Borrelia can tweak its gene expression so precisely, it can actually adapt to our defenses in real time. It's like it's reading our immune system's playbook and adjusting its strategy on the fly.
Okay, that is both incredibly cool and totally terrifying at the same time. So we've got this master manipulator, Borrelia, slipping past our defenses with its fancy gene regulation. But it gets even sneakier, right? Didn't you say it had, like, a whole bag of tricks for hiding from our immune system?
Absolutely. Remember how we talked about those plasmids being like USB drives with special tools?
Huh.
Well, some of those tools are specifically designed for, like, stealth mode.
Okay.
One of the sneakiest things Borrelia does is it actually lacks a typical outer coat that most bacteria have, called an LPS layer.
So it's like it's going incognito, ditching the usual, like, bacterial uniform.
Exactly. Most bacteria have this LPS layer. It acts like a big flashing neon sign screaming, "Hey, immune system, over here, come and get me."
Right.
But Borrelia's like, nope, not playing that game.
Smart move. But how does ditching the LPS layer help it exactly?
For starters, it makes Borrelia much harder to recognize. Our immune systems are really good at spotting patterns, like saying, hey, you don't belong here. But without that LPS, Borrelia can kind of blend into the background. It's like wearing camouflage in the jungle. You're just much harder to spot when you blend in with your surroundings.
Okay, that makes sense. But blending in can only get you so far, right? What happens when our immune system does catch a glimpse of this sneaky bacteria?
Well, Borrelia has a backup plan for that too, and it's called antigenic variation.
Antigenic what now?
Think of it like this. Imagine you're trying to find someone in a crowded room, and they keep changing their clothes.
Right.
One minute they're wearing a red shirt, the next it's blue, then green. It'd be pretty hard to keep track of them, right?
Yeah, that sounds super frustrating. So is Borrelia like a master of disguise? Changing outfits to confuse our immune system?
Exactly. It's constantly changing its surface proteins. Those are the things our immune system uses to identify invaders. It's like it's wearing a different hat every day so our immune system doesn't recognize it. The key to this whole costume change is a protein called VLSE.
VLSE. It sounds like something out of Star Wars. So how does this VLSE thing work?
It's pretty wild, actually. VSE is constantly mutating, changing its genetic code, just a little bit here and there. And each tiny change creates a slightly different version of the protein on Borrelia's surface.
So it's like it's shuffling its genetic deck of cards, creating endless combinations of disguises. No wonder our immune system has a hard time keeping up. But surely there has to be some way for our bodies to fight back, right?
You'd think so, but Borrelia has one more trick up its sleeve, something we call Core-ASP.
Okay, now you're just messing with me. Core-ASP, what in the world is that?
I know, it's a lot of acronyms.
Yeah.
But trust me, this one's important. Core-ASP stands for Complement Regulator Acquiring Surface Protein.
Okay, I'm going to need that translated into English, please.
Basically, Core-ASP acts like a shield. It allows Borrelia to grab onto molecules in our blood that normally help regulate our immune system, preventing it from going haywire.
So it's like Borrelia is hijacking our own defenses and using them against us.
Exactly. It's like it's saying, oh, you thought those immune cells were coming after me? Think again. They work for me now.
This is blowing my mind. So not only can Borrelia change its disguises, but it can also grab a hostage to use as a human shield.
You're getting the picture. But remember how we talked about Borrelia needing to adapt to survive in different environments?
Right.
It turns out it doesn't just change its outfit, it uses specific tools for specific jobs, particularly when it comes to moving around.
Okay, so we've got Borrelia, this master of disguise, rocking its camouflage, swapping outfits like a fashion show, even taking hostages from our own immune system. But I'm still a little fuzzy on how it actually gets around. It's got to move from the tick into my bloodstream and who knows where else.
You're right,
Yeah.
just surviving isn't enough. Borrelia has to be able to navigate, spread, set up shop in different parts of the body. And this is where things get even more interesting. We're diving into its impressive collection of proteins. Remember those specialized tools on the plasmids we talked about?
Yeah.
Some of them are like all access passes to different parts of the body.
Oh man, this just keeps getting more elaborate. What kind of all access passes are we talking about?
Well, our sources mentioned this family of proteins called OSPs.
Huh?
OSPs, yeah, two of them, OSPB and OSPB, are essential for that tick phase of Borrelia's life cycle. Think of them like specialized hiking boots, perfect for gripping onto that tick gut lining. Without them, Borrelia would have a tough time holding on and wouldn't be able to make the jump to us.
So if there were no OSPA or OSPB, that tick I found might not have been a Lyme risk at all.
Exactly. Those proteins are critical for transmission. No OSPA and OSPB, no Lyme disease, at least not from that tick bite.
That's both fascinating and a huge relief to hear. It's like those proteins are a weak link in the chain if we could target them somehow.
You're thinking like a scientist already. And there are actually researchers looking into that exact strategy. But of course, Borrelia has a few more tricks up its sleeve. It wouldn't be a master of adaptation if it only had one way to get around, or
Oh, I know this bacteria has backup plans for its backup plans. What else does it use?
well, remember when I mentioned that Borrelia needs to move through tissues to really spread?
Yeah.
There's another OSP protein, OSPC, that seems to be involved in that process.
Wait, so different OSPs for different environments? This bacteria is too smart for its own good. What does OSPC do?
This one is, well, a bit of a multitasker. Not only is it involved in that transmission from ticks to humans, but it also appears to help Borrelia move through our bodies by binding to a protein we produce called plasminogen.
Plasminogen? That sounds familiar. Doesn't that have something to do with blood clotting?
You've got a good memory. Yeah. Plasminogen is involved in breaking down clots, but it also plays a role in how our tissues are structured. And Borrelia seems to hijack plasminogen, using it to basically clear a path for itself.
Wow, so OSPC is like a bacterial battering ram powered by our own proteins. No wonder Lyme disease can affect so many different parts of the body.
Exactly. It's that ability to move and adapt that makes Lyme disease so complex. And, Borrelia has another trick to help it get around.
Oh no, not another one. What else could it possibly do?
This one's all about sticking to our tissues.
So it's not enough to just move around. It wants to set up shop and get comfy.
Exactly. And it's got just the tool for the job. These things called decorn binding proteins, DBPA and DBPB for short.
DBPA, DBPB. I'm starting to feel like I need a glossary just to keep up with all these proteins.
I hear you. But think of it this way. DBPA and DBPB are like grappling hooks that help Borrelia latch onto a protein called decorin, which is found in connective tissues all over our bodies.
So it's like Borrelia is Spider-Man swinging from tissue to tissue with its little grappling hooks.
You got it. That's a great way to picture it. And this really highlights the complexity of Lyme disease. It's not just about the bacteria themselves, but about that intricate dance between those bacteria, their proteins, and our own bodies. It's a constant back and forth, a molecular arms race.
This whole deep dive has been a wild ride. We've uncovered secret bacterial blueprints, witnessed some grand costume changes, even met some molecular grappling hooks. Seriously, this is a lot to process. What are the key takeaways here?
You're right. We've covered a lot of ground. I think the biggest takeaway is just how adaptable and resilient Borrelia really is. It has this remarkable ability to change its tools and tactics depending on its environment, which makes it a formidable opponent. And, that's why Lyme disease can be so challenging to diagnose and treat. It's not a one-size-fits-all situation.
Totally. We can't just throw antibiotics at it and hope for the best. We have to understand its tricks, which is exactly what we've tried to do today.
Exactly. And the more we learn about these intricate mechanisms, the better equipped we'll be to develop those new treatments and prevention strategies.
Well, I, for one, am feeling very grateful for all those researchers out there working tirelessly to unravel this complex disease. And I've definitely learned way more about Lyme disease today than I ever thought possible.
Me too. It's a fascinating field, and, there's still so much we don't know.
That's the beauty of science, always more to discover. But for now, I think we've all earned a break from the microbial espionage. To our listeners, thanks for joining us on this Lyme disease deep dive. We hope you enjoyed this journey into the fascinating and sometimes kind of unnerving world of Borrelia burgdorferi. Remember, knowledge is power, especially when it comes to our health. Stay curious, friends.